Let us consider the microscopic features of liver. I am grateful to Dr. Michael Horsch of University of Michigan Medical School for providing the images of histology sections. Liver is the largest internal organ located in the upper right abdomen. Except at the bare area and sites of attachment of peritoneal folds, it is entirely covered by serosa. Beneath the serosa, it is covered by a thin Glisson's capsule made of connective tissue, and this capsule sends in trabeculae carrying branches of portal vein, hepatic artery, and bile ducts called as portal tracts. Liver parenchyma consists of plates of hepatocytes that appear to be radiating from the central vein. These plates of hepatocytes are usually 1 cell thick in case of adults. When we see more than 2 cell thickness in adults, it indicates either regeneration of hepatocytes or malignant changes. Sinusoids are present between the plates of hepatocytes, and the gap between the endothelium of the sinusoids and the hepatocytes is the perisinusoidal space of Disse. We find cells of ito with reticulin fibers and microvilli of hepatocytes in this perisinusoidal space of Disse. Connective tissue stroma of the liver contains portal triads. We are seeing a magnified portal triad here, showing a branch of portal vein, hepatic artery, and interlobular bile duct, all of which are covered by a connective tissue sheath forming the portal canal. Between the portal canal and surrounding hepatocytes, there is periportal space of Moll. This forms the site for the beginning of lymphatic drainage of liver. Lymphatic vessels which begin here will also travel along with the portal triads. Unlike in pancreas, where the exocrine and endocrine functions are performed by two different sets of cells, in liver, both these functions are performed by the same cells, that is, hepatocytes. These hepatocytes secrete exocrine bile, they modify various endocrine hormones. In addition, they take part in carbohydrate, lipid, protein, and ion metabolism, synthesize various plasma proteins, store and modify various vitamins, and help in degrading drugs and toxins. Being the main type of cells in the liver, they make up 80% of liver volume, 60% of cell population, and have a lifespan of about 5 months. They are large polyhedral cells with 5 to 12 surfaces and having a size of about 20 to 30 micron diameter. They show 3 types of surfaces. Sinusoidal surface which face the sinusoids. Here they show presence of microvilli, and around the circumference, part of the opposing plasma membranes of adjacent hepatocytes are grooved, forming the bile canaliculi, and this surface is called as canalicular surface. Margins of these bile canaliculi are sealed off by tight junctions to form the blood-bile barrier so that the bile does not leak into either perisinusoidal space or the intercellular space. All the remaining surfaces where the hepatocyte comes in contact with the neighboring hepatocytes are labeled as intercellular surfaces. These cells show a round euchromatic nucleus with 2 to 3 nucleoli. Tetraploidy and binucleate cells are also commonly seen. Cytoplasm is eosinophilic due to the presence of smooth endoplasmic reticulum, Golgi apparatus, and numerous mitochondria, which suggest high metabolic activity. Basophilic stippling is seen here due to the presence of rough endoplasmic reticulum and free ribosomes, which are required for the synthesis of plasma proteins. They have a foamy appearance due to stored glycogen and vacuolated appearance due to lipid droplets. Hepatocytes also contain urease-containing vacuoles, ferritin and hemosiderin-containing vacuoles, numerous peroxisomes, and lysosomes with lipofuscin granules. Secretory vesicles containing bile are also seen close to the canalicular surface. Apart from hepatocytes, there are many other types of cells in the liver. Hepatic stellate cells or the ito cells are the irregular mesenchymal cells found in the space of Disse. They help in storing vitamin A. However, in case of liver injury, they transform into myofibroblasts and take part in repair process, forming the collagen scar. Kupffer cells are the hepatic macrophages. They are irregular cells with long processes seen in the internal surface of sinusoids. They form the part of mononuclear phagocyte system and help in removing the microbial debris in the sinusoids. Following splenectomy, they also help in phagocytosing old RBCs. We also find endothelial cells which are lining the sinusoids. These are fenestrated flattened cells with a central nucleus. Cholangiocytes are the cuboidal or columnar cells with apical microvilli, and these cells are found to be lining the biliary ducts. Stem cells are the undifferentiated pluripotent cells. These are found around the canals of herring. When they divide, they can form both hepatocytes and cholangiocytes. In addition to these cells, we find migratory blood cells passing through the sinusoids, pit cells or the natural killer lymphocytes, and various types of connective tissue cells. Structural arrangement in the liver is described in three ways, namely a hepatic lobule, a portal lobule, and a portal acinus. The hepatic lobule or the classic lobules are the hexagonal areas with a central vein at its center and portal triads at its vertices. This depicts the direction of the flow of blood through the sinusoids towards the central vein. Portal lobules are triangular areas marked by imaginary lines connecting three closest central veins around a portal triad. This emphasizes the exocrine function of liver, marking the area which is going to drain into one interlobular bile duct. Portal acinus is an ovoid or a lozenge-shaped functional area, which is marked by connecting two adjacent portal triads with two closest central veins. It correlates the blood supply to the liver with the metabolic activity of hepatocytes as well as certain pathological conditions. So here we are seeing a section of liver showing the hepatic lobules. We are also seeing one hepatic lobule being marked by this line. In some species, hepatic lobules have a well-marked connective tissue boundaries. However, such arrangement is not seen in case of human beings. Liver receives its blood supply from dual sources. 75% of its blood supply coming from portal vein carrying the nutrients and toxins from the intestines. 25% is coming from the hepatic artery carrying the oxygenated blood. At portal triads, the branches of portal vein and hepatic artery open into the sinusoids, and within the sinusoids, the blood flows towards the central vein. Despite its various activities, hepatocytes never get fully oxygenated blood. Instead, they receive this mixed blood coming from both portal vein and hepatic artery. In a portal acinus, as shown in this schematic diagram here, the hepatocytes are described to be arranged in three different zones. Zone 1 or the periportal zone has the cells which are closest to the portal triads. Zone 2 is intermediate in position, and zone 3 has the cells which are closest to the central vein and hence are the last to receive the sinusoidal blood. Although the cells in zone 1 are the first to receive nutrients and oxygen, they are also the first to receive toxins and hence are the first cells to undergo toxic necrosis. These cells are also the first to be affected if there is obstruction to the bile flow. Cells in zone 3 are the last to receive blood from the sinusoids and hence are the first to show ischemic changes in case of decreased perfusion. Cells in zone 2 show intermediate responses. This is a section of liver showing a portal acinus and zones 1, 2 and 3. Let us now consider the pathway of the bile. Bile is synthesized by hepatocytes and is secreted into biliary canaliculus, which is formed by opposing grooved plasma membranes of the adjacent hepatocytes. Bile is then drained into intralobular canals of herring, which are lined by both hepatocytes as well as cholangiocytes. Canals of herring drain the bile into periportal biliary ductules, which are exclusively lined by short cholangiocytes with apical microvilli. Biliary ductules then drain into interlobular bile ducts, which are found within the portal triads. The smaller interlobular ducts are lined by cuboidal cholangiocytes, whereas the larger ones are lined by columnar cells. Interlobular ducts join together to form right and left hepatic ducts, which join with each other to form the common hepatic duct. Bile from common hepatic duct drains to and from the gallbladder via the cystic duct, and distal to cystic duct, the common bile duct finally releases the bile into second part of duodenum. Here we are seeing cholangiocytes lining the bile ductules near the portal triad. We are also seeing cuboidal cholangiocytes lining the interlobular bile duct in a portal triad. Here we are seeing columnar cholangiocytes lining the larger interlobular bile duct. So quickly recollecting what we have seen so far, liver parenchyma is made of plates of hepatocytes interspersed with sinusoids. Glissen's capsule sends in trabeculae with portal tracts containing branches of portal vein, hepatic artery, and bile ducts. In addition to hepatocytes, liver also contains itocells, Kupffer cells, endothelial cells, cholangiocytes, stem cells, and various blood and connective tissue cells. Structural organization in the liver is described as a hepatic lobule based on the direction of blood flow, a portal lobule based on the direction of flow of bile, and a portal acinus correlating the blood supply with metabolic activities. Direction of flow of bile is opposite to that of blood, that is, it starts from the hepatocytes and flows towards the porta hepatis. Thank you. Hope you enjoyed this video. You can also visit this site for similar histology videos.